tardes a todos, muchas gracias por venir. Buenas tardes también a Madrid. I'm going to talk in English because Professor Pikarsky is coming from Israel. He's the chairman of the pathology department in the Hebrew University. He's also a chairman for different associations of cancer research in Israel and also in Europe. And he's also involved in the mechanism of tumor progression, inflammation, and cell biology in cancer. So, as you may know, he's a big name in this area, immunology and HCC. And it's a big pleasure to introduce him and share with him this session. Thank you very much for coming.
So thanks a lot, Maria and Jordi, for inviting me to this very beautiful city, and more importantly, organizing a really exciting day. It's really been a pleasure to interact with a lot of you, to learn what you're doing, and hopefully to generate new connections and interactions. Maybe one correction, I'm not the chairman of the European Association for Cancer Research. I used to be a member of their board, but I'm not anymore. This is...
For me, you are.
Well, I don't want to be. We discussed yesterday, you need to give away power. It's very important, so other people will do better things. So I'm gonna now switch to science. I'm better at science than making jokes, hopefully, which doesn't mean that I'm a very good scientist, but that was a joke. Anyway, so I'm going to talk about. I'm going to try to talk about the immunology of HCC. I'm going to start the lecture with some general comments on how this field is viewed. Somebody is waving at me as if I'm not doing something correctly. Everything is okay?
Yeah, don't worry.
I'm a worried person. That's why I'm a pathologist.
I'm just doing it.
Okay, thank you. So, and I think what is important to know is that the field of immunology in general, and maybe more importantly of immunology of specific organs, is changing. It's a notion that we didn't really accept until very recently, but we're learning more and more that the function of the immune cells is changing when they become part of a specific organ. They are much more complex and complicated than we tend to think of them. So we oversimplify things. So that's one thing. And the other thing is just saying that it is all about balance. Everything in our life is about balance. We want to balance ourselves, balance everything in our life, which is one of the most difficult tasks. But the immune system, the balance is extremely important, and it's the balance between two sides. What we call protective immunity. So these are the good sides of the immune system, which is here to protect us against pathogens, to control physiology, to restore homeostasis. But there is also immunopathology. And immunopathology is underlying the most important diseases in humans, like cancer, like atherosclerosis, probably also Alzheimer's disease, diabetes. So this immunopathology is what you want to prevent, and protective immunity is what you want to achieve. But because this system is so complex, while it is a very robust system, it's robust because it can adapt and help the organism to resist many different pathogens and attacks. However, we know that in many occasions overactivity or underactivity of the immune system results in pathology. And so if you realize this, then at least my life as a scientist is very simple. I need to sort out what causes protective immunity, what causes immunopathology, and then try to restore the balance, to heighten the protective immunity. But it sounds simple, but it's not simple. So like everybody that is using the new immuno-oncology drugs knows that side effects are side effects of overactivity of the immune system. So this is, when you think about it, think about this balance between what is protective and what is pathological, but also remember that what is protective for one disease is going to be pathological for the other. So everything, you can't think of yourself as an immunologist today without talking about immune checkpoint inhibitors. And I didn't write it, but I think I can say it out loud. When we talk about immune checkpoint inhibitors, most of what is working in the clinic is inhibiting the PD-1, PD-L1 axis, and which directly affects the cytotoxic T cells. And the Nobel Prize that was given in medicine, that was given last year, goes for that, and I think everybody expected it. It's really a revolution in treating cancer. The brave oncologists today are talking out loud about curing metastatic cancer, something that we never had before. And maybe it's too early, but I really think that we are observing cures for metastatic disease, which was never achieved beforehand. But now, when you think about the different tumors, there is a huge difference. And let's talk about the 3 tumors which we know best with regards to the response to the PD-1 inhibitors, which are melanoma, colorectal cancer, and lung. So in melanoma, most patients respond. The notion is that they respond because of the high mutational load and the neoantigens that are associated with it. But still, still patients are dying with melanoma, so we don't really understand it. So far, we don't have any good predictive biomarkers for the response in melanoma, even though there are several papers out there. Maybe the most important ones are those that are testing the phenotype of the tumor infiltrating lymphocytes. So, and I want to stress this, it's not enough to say what is the number of lymphocytes and what is the type of the lymphocytes that are present in the tumor. You probably need to assess the exact phenotype of the lymphocytes, and this is becoming easier and easier now with time. In colorectal cancer, life is much more simple. The patients that have mismatch repair deficiency and therefore have high mutational load respond dramatically to the immune checkpoint inhibitors. All other patients do not respond, regardless of the level of expression of PD-L1, regardless of tumor infiltrating lymphocytes, and so on. And in the lung, we have another biomarker, which is expression of PD-L1 by tumor cells. And our lives, I'm a pathologist, and our lives is becoming more complicated because we need to generate and to assimilate and incorporate into our clinical decision-making, pathological decision-making, more and more biomarkers that predict response to immune checkpoint drugs. But I want you to stop and think back, and thinking sometimes is good. And why is there such a difference? I mean, why does expression of PD-L1 in the lung correspond, I wouldn't say very nicely, but nicely with the response to immune checkpoint inhibitors, but in the colon it does not predict at all the response? So you can think of different reasons for that. One would be a different mutational load, but we know that this is not really the case because this was checked. To me, this signifies that the immune system in the different organs is inherently different, because otherwise we wouldn't have seen this. And if this is correct, that would mean that for any organ, we first need to understand how the immune system controls tumorigenesis in this organ, and once we understand that, we will be able so this will be important not for generating better biomarkers, because biomarkers are important, but the real question I think today in the field of immuno-oncology is how can we increase the number of patients that are responding to treatment. And recently I asked one of my general oncologist friends, what are patients responding to most? If you take all cancer patients, so there is chemotherapy, immunotherapy, and targeted therapy against signaling factors. So most people would say, well, immuno-oncology and targeted therapy are improving the lives of more patients, but this is not the case. Chemotherapy is still the working horse of oncology, and we need to increase the number of patients that are responding to immunotherapy. And I think that if we understand all the factors that are taking place in a specific organ, and organs are still what we need to look at this stage of life, and if we understand the immunology, maybe we can say, what do you need to target in addition to PD-1 in order to kill a tumor cell? So I'm sure you all know this much better than I, but for us, the reason to start looking at more carefully at the adaptive immune system are the preliminary results that there is a significant response rate to immune checkpoint inhibitors. As far as I know, there is no phase 3 trial, but these trials are ongoing, and everybody you talk to say, well, we've seen complete responses, but in around 20% of the patient. We've seen responses in like a significant proportion of the patients, anywhere between 10 to 30%, depending on how optimistic the person you're talking to is, and some complete responses. So definitely there is room for activating the adaptive immune system in HCC, but we don't know to do it in all the patients. In this series, it was 17% of the patients which showed an objective response, which in HCC is very good. So let's talk about the immune system. So this is what we teach in medical school, and really, you know, it's not that I had to look hard to find a picture of the organs of the immune system which misses the most important immune organ in our body, which is situated here, and not surprisingly is the liver, because we think about the liver as a metabolic organ, and definitely the liver is a metabolic organ. It performs more than 500 biochemical functions, but it's the organ in our body which contains the highest number of immune cells. So you can't really neglect it when you talk about the immune system, much more than the spleen that anybody you talk to would think is the largest immunological organ. But it also plays very important functions. As written here, it is constantly regulated by the flux of DAMPs, damage-associated molecular patterns, and MAMPs, microbe-associated molecular patterns, which are present at very high levels in the portal blood. And the liver also actively performs constantly immunological functions. It is probably the largest organ that patrols the blood, controls the blood and removes bacteria. This is the work of the liver sinusoidal liver sinusoidal endothelial cells, but there are also dendritic cells and many other leukocytes. NKT cells are most commonly found in the liver. We don't really know what they do. We just have a name for them, and we define them as cells that express NKG2D and CD3, but we don't know their function. And the liver has multiple cytokines which have both pro-inflammatory and anti-inflammatory roles. Now, I think that I think I said all of this, and so the liver is an immune organ. Okay, it functions as an integral part, as a major integral part of the immune system. And now, if we want to simplify things, so I went a bit into the picture and mentioned many types of immunological cells and many types of cytokines, but in general, the liver is an immunosuppressed organ. So it's, I would say, partly immune privileged. And the reason for that is that the liver is constantly exposed to pro-inflammatory signals that come from the gut through the portal circulation. And it's not even a sterile organ as we thought classically, because if you look very carefully using high throughput genomics, you find bacteria in any normal liver. So how is this changing the activity of the immune system in the liver? And before we go into this, I want to show you results from Miriam Merad's lab in Mount Sinai in New York, in collaboration with Ido Amit at the Weizmann Institute. And what they did is they profiled the macrophages in different tissues, and you can see that while we give the cells the same name, we call them macrophages, they are not the same cells. The pathologists that describe them, they look similar to them, they have the same morphology, and they do have shared activities and expression patterns and functions, but they also have very distinct, unique roles. And what this paper shows, it not only gives this map of macrophage phenotype, and it also gives the reason, a possible reason. I'm not sure that this is strongly proven, but the data really imply that they found specific transcription factors which, when active in a specific macrophage subtype, like SMADs in the brain and PPAR gamma in the lungs, and GATA6 in the peritoneal cavity, they dictate the function of the macrophages. And these transcription factors are activated by resident tissue cells. And what is even more important is that this causes a permanent epigenetic switch in the phenotype of the macrophages. And when I'm saying permanent, I don't think it's lifelong. You know, macrophages can live for years in our body. Assumingly this can change, and probably within the tumor microenvironment these things do change, but there is a permanent memory to this which is written in the epigenetic makeup of the macrophage. And in return to this educational system, the macrophages then play specific roles in specific tissue. For example, the macrophages in the brain secrete IGF-1 and BDNF, which are critically important for the survival and growth of new axons. So the picture, what I'm trying to convey is that the picture is very complicated, but if we think about specific organs and try to learn the immune function of that organ, maybe we can begin to understand the complexity. And why is this important for tumor biology? Because when a tumor begins to grow, it grows in a very structured and constant immune environment. And this is the immune environment that it remembers throughout its life. Because if you're not adapted to the immune system in the organ in which you grew up, in which you were born, you won't survive. It's clear that the tumor will eventually progress and evolve, and during its progression and evolvement will cause a change, a shift in the immune system. But what would be common to tumors of the same organ is going to be their ability to adapt to that specific organ's immune system. Now, this is a hypothesis. I might be wrong, I might be right, but I think that, this is an inherent function of specific tumors. And again, I urge you to think, we know that we treat different tumor types with different chemotherapies today. And we know that they work differently. It's not just by chance. A specific drug combination, I'm not talking about targeted therapy, I'm talking about chemotherapy. A specific drug combinations that work for one organ system does not work for another. This is not trivial. There is a meaning to that which we don't know. And one possible meaning is, at least in my mind, that these tumors were formed in an environment, in a specific immune environment, and specific drugs can alter the activity of the immune system differently. Maybe, and this is really speculation. Maybe this is one of the answers. Now, what happens in HCC? So HCC is even more complicated. It's really a daunting task to understand the immunology of HCC, because most HCCs, 90% of them, around 90%, develop on the background of a chronically inflamed liver. So the immune system which the tumor was adapted to when it in its beginning, when it was born, is not the classical immune system in the liver, but rather the immune system of a chronically inflamed liver. And this makes the picture more complex, because my original hypothesis or message was that there is a constant and very similar between different human beings immune system in each organ, and a tumor evolving in this organ must adapt to this immune system. But if in the liver you first develop chronic inflammation and then you develop a tumor, then we're going to have different tumors developing in the context of different chronic inflammatory stimuli. And I think that this is indeed what we're seeing. The tumors that are evolving, the immunological makeup of tumors that are evolving with hepatitis C virus-induced HCC are very different than tumors that are developing in the background of NASH-induced HCC. So we need to divide the question to sub-questions, and that is, what are the immunological changes that are occurring in NASH? What are the immunological changes that are occurring in HCV? And then we could understand how to manipulate the immune system and try to increase the response rate to immune checkpoint inhibitors. This is a very complicated figure, which I'm partly responsible for. To be honest, I don't think it teaches me a lot, but I think what it does convey very nicely is that this is a very complex picture which we don't really understand so well. So, and obviously the reviewers agreed that this should be published, and but really, you know, it's too complex for us to understand it at this moment. And what really we have to tease out from the system is, of all of these interactions that are taking place, which are the important ones. And eventually there's only going to be one way to know which are the important ones, and this is going to be in clinical trials in human beings. Nobody could have told you 10 years ago that anti-PD-1 drugs are going to be much more successful than anti-CTLA-4 drugs, which are partly successful and hugely more successful than LAG-3, TIM-3, and all the other immune checkpoint drugs, which on their own currently do nothing. Okay? So why does PD-1 inhibition work better? Nobody really knows. But if you look at this, we can't really do prospective trials with all the potential combinations of drugs which you want to give to human beings. There's no way that we can ever do that. There is not sufficient money, not sufficient patience, not sufficient time. So we need to try to identify which are the important effectors from the non-important effectors, and this is actually the picture that we know today. And we really don't know which are the important players. And maybe even more importantly, how can we identify the ones that are going to be synergistic? Because this is what we're looking for. We're looking for a synergistic effect, not for an additive effect. And this takes us back to understanding this balance between protective immunity and immunopathology, and this is what we're trying to do in the lab. I think I've discussed this already. So the way that in the lab we are trying to begin to solve this problem is, so we use mouse models. And I think that at start I would want to say that mouse models are not perfect, are far from perfect. While there are many similarities between the immune system of humans and mice, there are differences. We are exposed, the evolution has selected for different functions of the immune system in different organisms. But if you look systemically on the immune system of a mouse and the immune system of a human, and there are now articles that show it, they are very similar. And when you look at the histology of immune reactions, they're actually remarkably similar between the two organisms. So I think, yeah, we can learn something from mouse models, but admittedly, obviously, we don't know everything, everything. So I think that in my mind, what you want to achieve is, you want to achieve some information from the mouse. Then you want to look what is happening in human tissues. Are you seeing something similar to what you see in the mouse occurring in the human? And if this is sufficiently encouraging, go and do a clinical trial, most of which are going to fail, but maybe it will increase the likelihood of success in clinical trials. And currently our success in clinical trials is not great. So I'm going to go now into specific examples, and this is one of our first attempts to understand the role of the immune system in liver cancer. And what we did here is we used a mouse model of chronic inflammation. And basically these mice, the MDR2 knockout mice, develop liver tumors at an annoyingly slow rate. About 1 year you have a 90% penetrance. Not great for a PhD student, but still an important model. And without addition of any exogenous stimulus, you see the appearance of tumors, and here we followed the mice with MRI. At that time it was still for free. Now we have to pay for it, so we stopped doing these MRIs. And the reason that the mice have chronic inflammation is because there is a defective bile transporter, and the bile is acidic, and this acid bile induces a portal inflammatory process, and this causes constant damage to the liver. And this is sufficient to induce tumorigenesis. Humans, some humans, very few actually, also have these mutations. We have a village in the north of Israel where we have families with this mutation, and few of them develop HCCs. We have one case. So mice with the same mutation human with the same mutation are more resistant. But this is true, I think, for every tumor model. Mice are more prone to develop tumors. This is one problem with our mouse models. It's very easy to generate cancer in a mouse. It's also quite easy to cure cancer in a mouse. And I think it has to do one with the other. So this is like a classical model to study chronic inflammation-induced hepatocellular carcinoma. And the dogma at that time, this was published I think 14 years ago or so. The dogma at that time was that chronic inflammation causes cancer by the generation of reactive oxygen and nitrogen species, and that chronic inflammation is mutagenic in this respect. And I like to think about cancer, again, to simplify things, to talk about initiators, which are factors which cause mutations, and promoters, which we don't really understand, but definitely one aspect of tumor promotion is protection against the immune system. Others, as I'm going to show you here, are evasion of apoptosis of any other means. You know, it doesn't have to be apoptosis induced by the immune system. So what we showed here, we looked for factors which could be activated in hepatocytes by the by the constant inflammatory reaction, and I'm going to show you again and again. You know, NF-kappa B is a transcription factor, which is a major regulator of innate immune responses, maybe the major regulator of innate immune responses. And in many questions which I ask, the answer is NF-kappa B, for better or for worse. So this was one of the first examples, and actually not the first one. But here again, NF-kappa B was a very simple answer to the question, and the hypothesis was that the inflammatory microenvironment releases cytokines which activate NF-kappa B in the hepatocytes, and NF-kappa B, and it was really known that cytokines can activate NF-kappa B at that time, and it was also known that NF-kappa B is anti-apoptotic. So the notion was very simple. Chronic inflammation induces high levels of cytokines which upregulate NF-kappa B in the hepatocytes. This upregulation protects transforming hepatocytes from tumor killing, either by intrinsic apoptosis or immune system mediated apoptosis, and this temporary prevention just allows a sufficient number of hepatocytes, of transforming hepatocytes, to survive to the next step, which gives them a cell-autonomous ability to avoid these tumor suppressor mechanisms. And we use the mouse model of inducible NF-kappa B inhibition to show that indeed when you inhibit NF-kappa B, you dramatically reduce tumorigenesis. We identified TNF-alpha as the major mediator of this NF-kappa B activation. And we drew this model, and you see that already then we thought about balancing. Since then I constantly try to balance myself very unsuccessfully. But what you can see is we identified a single molecule, we identified a single responder, And you need to do this if you want to publish a paper in a reasonable journal. But this is wrong. And this is one of the problems of science today. We are forced to simplify the picture, and to be honest, when we published this, we already knew that we are giving a much too central role to TNF-alpha in this system. NF-kappa B is a central player, but it is activated by multiple other cytokines. And since then, in many other systems, many people have shown that additional cytokines and additional inflammatory signaling pathways play the same role. So one way through which inflammation promotes tumorigenesis is by changing the phenotype of the transforming cells and protecting them from various gatekeepers, be it apoptosis, adaptive immunity, innate immunity mediated killing, and also multiple cytokines were later shown to do the same work that we have shown for TNF, like lymphotoxin beta, which was once called the TNF beta by chance, and IL-6 and HGF and multiple other cytokines. So the next question that we try to address is, okay, so we know that these macrophages are very, I forgot to say before, but we knew that macrophages are the source of the TNF in our system, in the chronically inflamed liver, and also the source of IL-6 and other pro-tumorigenic cytokines, which were shown by others to be pro-tumorigenic in the setup of chronic inflammation-induced hepatocellular carcinoma. So at this time it was beginning to emerge that the adaptive immune system is playing a critical role in protection against cancer. And the young people in the audience think that we always believed so, but I can tell you that 15 years ago in some places saying that the adaptive immune system controls cancer was considered a joke. It's really interesting to look at the history of the development of this science. Today, it is very clear that all of these immuno-oncology drugs are working by activating the adaptive immune system. So what we did is we looked for a connection between the innate and the adaptive immune system. And again, this is a simplification, and I realize that it is a simplification because practically the fact that we call them an innate and an adaptive immune system is wrong, right? Every cell in our body is a member of the innate immune system, and the fact every cell in our body can activate NF-kappa B and can summon an adaptive immune response. And in this respect, every cell in our body participates in adaptive immune responses. But still, we have to simplify the picture in order to generate insights. And what we looked for are signals that are derived from these innate immune cells, which we know accumulate in chronic hepatitis, and recruit immunosuppressive cells into the liver, and allegedly, we didn't prove that, allow tumorigenesis to proceed. And again, it's a kind of a proof of concept study, and what we showed is that a specific phosphorylation event that is occurring in the macrophages, and for some reason takes time to exert its effect. And when I'm saying takes time, about 3 months. So while the macrophages are exposed to this, same, more or less the same cytokine milieu, for a long period, it takes several months until this phosphorylation event affects their phenotype. But if you completely remove the specific serines that are phosphorylated in this case, they cannot make this transition from anti-tumorigenic to pro-tumorigenic macrophages. But when they do so, they secrete cytokines which recruit T regulatory cells, which are well known in multiple human studies. And I just saw today another example which proves again that T regulatory cells are associated with a bad prognosis in HCC. In fact, I think that the fact that T regulatory cells correlate so well with the prognosis in HCC, for me, is a sign that there is a good chance that we can, if we understand the immune system in the liver better, we can get better results with immuno-oncology drugs. And in the last 10 minutes or so, I want to draw. So you saw that we started with studying the macrophages, which are classical innate immune cells, and then we studied the connection between the innate and the adaptive immune system, and we couldn't resist in recent years to go to purely adaptive immune system functions, simply because all the world is going there, and we were swapped by the current of people moving to the adaptive immune system. In this case, as a pathologist, you often see these lymph node-like structures, and here is one that really looks like a lymph node if you remember your histology lessons, but it's not a lymph node because it is present in the liver. And this is a tertiary lymphoid structure. There are many names for them: tertiary lymphoid organs, ectopic lymphoid structures. From now on, I'm going to refer to them as ELSs. And every one of us in this room has ELSs in our body, and they develop in most chronic inflammatory conditions. In some diseases, they are pathognomonic, like we don't diagnose Hashimoto's thyroiditis without the presence of these ELSs, and in some diseases, they are used as markers of treatment failure in this case, for instance, in transplant rejection. And it is not by chance that both these diseases are diseases which are associated with the immunopathology part of the balance between protective immunity and immunopathology. So we don't know a lot about these microorgans, as I like to call them, but they are thought to be the frontal command posts of the immune system. So while we think of the immune system as a systemic system, as I told you before, there is a very important organ-specific or local effect of the immune system, and there likely is an advantage of generating a local adaptive immune response. And to generate a local adaptive immune response, you want to generate these microorgans. And so I told you that we see them in thyroiditis and in transplant rejection, and in many other diseases where they are thought to be pathologic, to support immunopathology. In cancer, they were regarded as protective organs because of their supposed roles as effectors of an adaptive anti-tumor response. And in many tumor types, the presence of these ELSs is associated with a better prognosis, and Jim Moulet from the Moffitt Cancer Center was, I think, the pioneer in studying these ELSs, and his group and many groups later on showed that ELSs in colorectal cancer and in melanoma are associated with a good prognosis. Actually, in melanoma today, in our pathological reports, we write about their presence, and today also in colorectal cancer, because their presence is associated with the mismatch repair deficiency phenotype. So so it can suggest the the pathogenesis of the disease. I think I've shown this already. And so what about liver cancer? And to study the role of these ELSs in liver cancer, we collaborated with Eugene Hoshida. And I'm talking now about the ELSs in the liver parenchyma. And you see that they have a very dramatic effect, but a counterintuitive effect. And basically, the patients with a high ELS score have a higher likelihood of recurrence, and this is counterintuitive. And since then, the same notion was identified in other tumor types, very few, admittedly, like transitional cell carcinoma of the bladder and subtypes of breast cancer. So to study this, I told you what we do is we develop mouse models, and we developed a mouse model which generates the formation of these ELSs, and we were very happy to see that if you follow these mice for a sufficient time, they also develop tumors. So there is this, we can recapitulate this correlation between high number of ELSs and tumorogenesis. And what was more interesting, I'm sorry, maybe, is the fact that I'm missing a slide here. I'm sorry. That when you look under the microscope, and this is something that I urge you to do more than you are doing today, it can be very revealing. The microscope. You don't agree with me?
I agree.
You agree. Thank you. It was very obvious that the association between these two was not only an association, but there was a spatial event that constantly repeated itself, and that is that the earliest HCC progenitor cells that we could identify in the mouse liver appeared inside the ELSs. And Shlomi, who did this project, urged us to look at the humans. I told you at the beginning of the lecture that this is what you have to do. And I was afraid to do it because I said, well, we never see this. And he says, well, you never see it because you don't look for it. And they were very easy to find. So you stain an ELS in a human liver, and then you do fluorescent markers for HCC progenitor markers. And it's very easy to see these cells which have many attributes of HCC progenitor cells. We never proved that in humans that these are HCC progenitor cells within these lymphoid follicles. So suggesting that there is some sort of interaction between the immune follicles and the HCC progenitor cells, and to tease out whether the ELSs are trying to kill the tumor cells or they're helping them to grow in a manner similar to what I've shown you before with the macrophages. We just eliminated the adaptive immune system altogether, and this showed very clearly that when you eliminate the ELSs, you markedly reduce tumorogenesis in this model. And we went on to show that this is due to the provision of cytokines, and again we pointed to a specific one, but I'm sure that there are multiple others. So we identified lymphotoxin beta, but to be honest, I'm sure that it won't be sufficient to inhibit lymphotoxin beta in a clinical setting where tumors develop on the background of ELSs. The picture is more complex than what we have shown in that paper. Since then, there are two manuscripts out which make the system even more complicated. The first came from Jessica Zuckman-Rossi's group, Julian Calderaro, who is a pathologist. They looked in general in HCC, and they showed that ELSs that are present in the tumor, not in the parenchyma, are associated with a good prognosis, with a lower risk of recurrence after surgery. But this is not surprising, because the ELSs in the tumors are a response to the tumor. They're not a response to the chronic inflammatory process that is occurring in the liver parenchyma. So you would expect this. But even more annoying, I must admit, is the fact that Herb Friedman has looked at HBV-associated HCC, and there the correlation is completely the opposite of what we found. So higher numbers of VLSs in HBV-associated HCC is again associated with a good prognosis. And this cohort that we looked at together with Eugene, there is a high representation of HCV patients, and I can tell you, we looked now at the data, all the patients which we classified as high ELS score are associated with HCV. So ELSs which are formed on the background of HCV are bad, but ELSs that are formed on the background of hepatitis B virus and probably also NAFLD are good ELSs. So it's not the liver which makes the ELSs bad or good. It's the specific chronic inflammatory state which is occurring in the specific patients. And it's, I think, interesting to note that the reason that HCV is pro-inflammatory is because it constitutively activates NF-kappa B, so it's very similar to the mouse model which we used, and we see much less activation of NF-kappa B in HBV-associated hepatitis and also in NAFLD. I'm not talking about the tumor, I'm talking about the parenchyma. So these ELSs can form, can generate, can be either bad or good. I'm going to try not to run and just show you the important data. So we have good ELSs and bad ELSs. And can we distinguish them? And I think that the most important thing in this respect is, can we convert a bad ELS into a good ELS? Even if we're able in the mice to convert good ones to bad ones, it's good because it will teach us on their biology, and then maybe we can do it better in the patients. So to begin to answer this, and I don't have an answer, we just have theories which are partly proven and are still on a very weak basis. We wanted to find a way to characterize them in a very comprehensive way, and we tried several methods, but the one that did give us an answer in this case is single-cell RNA sequencing. And what you're seeing here is the immune it's a map of the immune compositions. These are these TSNE plots, and basically what you do is you isolate the immune cells from whatever you want to study, and you generate the RNA sequencing for each cell. It sounds very complicated, but technically it's a very simple procedure. The problem comes when you need to analyze the data. But basically what we did here is we compared the immune systems, the composition of the immune system in the spleen and in the ELSs, and you can see here that one of the cell components that we see here are these CD8 cells which are present in this population. There are other populations of CD8 cells, but this specific population is the one that is most interesting. And what was interesting about this population is the fact that the T cells, this specific population, showed signs of T cell exhaustion. And you can see here in this analysis that these cells express PD-1 because exhausted T cells express PD-1, but also activated T cells express it. But there is low levels of interferon gamma compared to the activated T cells. So and we proved this using immunofluorescence analysis, which is a cheaper but slower way to prove your point. And this shows sources for PDL1. and then we tried, and then I'll skip this, and we did the experiment that everybody expects you to do today, and we gave, we inhibited PD-1 in this system. And while we do see this mildly significant effect, it's not a strong effect. We expected much more from the system, because you have exhausted T cells. You expect that you will be able to reinvigorate them, because you see the difference only in the larger tumors. So most of the tumors, there is no difference whatsoever. So we think we identified exhausted T cells as a marker of the bad ELSs, but trying to reinvigorate them was so far unsuccessful. So I want to leave a very short time, I'm sorry, for questions, and maybe just show one additional result, and that is and maybe this is this is the beginning of an answer. Why are there more exhausted T cells, and why do we see, what is the source of the bad function of the ELSs? And we did several types of experiments here, and a lot of guesswork, you only see the experiments that did work, and while trying to understand the sources of the ELSs in the mouse liver, we use mice that are knocked out for ROR-C, so they don't have innate lymphoid cells, which are cells which are known to be critical for forming ELSs and also lymph nodes and spleens. So these mice don't have lymph nodes, they don't have spleens, and they're supposed to develop less ELSs. But when we crossed the IKK mice with the ROR-C knockout mice, we saw a dramatic increase in the number of ELSs, and this is the result that you like, right? You see a tenfold increase in something, and it's unexpected. It's not what you hypothesized, at least not what I hypothesized. But more interestingly than the increase in the number is the fact that we saw a reduction in tumorogenesis. So you see more ELSs, and these ELSs are behaving better. And it was very rewarding so we repeated this single cell analysis, and in this case you see, so these are all the CD8 T cells, they are marked here, and in this picture you're seeing the cells that were isolated from the ROR-C knockout mice, and the controls, which are heterozygous for ROR-C, and you see that there is a similar number of CD8 T cells in all these populations. It's this population of CD8 cells which is present in higher numbers in these good ELSs. And again, this population in this case are activated T cells. So we think that this is the beginning of the story. So the attraction of innate lymphoid cells into the ELS causes this immunosuppression in this case. So the exact cells that you would expect to generate a strong adaptive immune response, in this case suppress the immune response. And I'll skip all this, and I want to thank the many people that worked on this. Einat is doing the single cell analysis. Ilan is my lab manager and actually my boss for many years. He controls my life and does it in a very good way. Shlomi Finken started the ELS project, and this was and we're collaborating a lot with Matthias in Heidelberg, with Michael Karin on this IKK project with Eugene, and Oren helped us with the single cell analysis, and we have to thank everybody that funds our business, which is quite expensive. We like to spend a lot of money. And I will be very happy to take questions if Maria will allow a few minutes. I completely agree with you. I like when things are vague. We don't have a good definition of an exhausted T cell. I think so far the strongest way to define a T cell as exhausted is testing it in vitro and see its proliferative capacity in response to pro-proliferative signals. There is a study in melanoma that was recently published in Cell, where they used multiple technologies, including single-cell sequencing of tumor-infiltrating CD8 T cells in melanoma, and they identified a specific transcription factor, I think TCF8, which they claim is inducing the exhausted phenotype in tumor-infiltrating cells. I don't know if this occurs in HCC, but this could be a potential marker. I think that we are trying to categorize cells in a simple way, and we are bound to fail because cells are not simple. But we have to succeed somehow. Yes, well, so the question is, are the circulating immune cells going to help us? So I think I would divide this question into two. The first is, are the peripheral blood cells a surrogate marker of what is happening in the liver? I think the answer to that is no. We know from mouse models, and we should know very soon from human studies which are in development, that people are trying to do it because to see, can we find something that is indicative of what is happening inside the liver? I think the answer is no, because the system is too complicated, and you cannot tease out the cells that are coming from the liver from the rest of the cells. But the other question is, can we generate a systemic understanding of, so is the systemic response indicative of the ability of the patient to activate or suppress his or her immune system? To this, I think the answer is going to be yes, but we are very far from it. So yes, there is a value of assessing the systemic immune response, but mostly in addressing the general ability of the immune system to respond, not as a surrogate marker to what is happening in the tumor itself. So in MDR2 knockout mice, we do see cholangiocarcinoma from time to time. But trust me, we looked very carefully at the MDR2 knockout mice, and they don't have ELSs. Even, we have now mixed them with many genetic things, and no ELSs there. In the IKK mice, which I showed later on, a lot of the tumors which we see are mixed HCCs and intrahepatic cholangiocarcinomas. And I think, so our preliminary data, it's not very strong, but it's kind of repeating itself, is that the ELSs are, the bad ELSs are more supportive of CCC, or are more critical for CCC than for HCC. You can speculate about the causes for it. One possible cause is the fact that when you do a genomic analysis of these tumors, the CCCs have a more complex genome, so maybe they have more neoantigens. But we don't really know that. I think that what Eugene also analyzed the chances of recurrence of disease and not just outcome. So this would be one answer. But even so, this is the problem with the human model. It's dirty. Yeah. So the reason mice are effective is because you have this very clean system, and we had the same results in Jerusalem and in San Diego in Michael Karin's lab, and now there is a lab in Dublin, I think, that are generating the same results with another model of activating ELSs. So I think it's there. I think it's illuminating something. If eventually we can cure cardiac transplant rejection with it, I will be happy. The honest answer is that I don't know. I think they're coming from circulating lymphocytes, but this is guesswork. We tried to answer this, but so far unsuccessfully so. Well, the definition of a good question is something that I thought about before. So this is a very good question, but for which I do not have an answer. I think it's a very good point. I think we've discussed this. I think that the DAA story really provokes the notion that there is some pathological immunity that is taking place here. And my currently favored pathological immune response or bad DLSs. Doesn't mean that this is the answer, but it's certainly, and it's easy to test. We just need the biopsies. We need to biopsy more patients.
Thank you very much. Do you have more questions? Okay, thank you very much again, and see you next time.